glad you could join me today as we're going to dive into the fascinating world of soil microbial ecology. I'm about to show you how this deep learning cool technology is shaking things up in this field, making easier and more exciting to explore those tiny ecosystems that shape our world. My background is mechanical engineering, you may be wondering, but I work closely with soil biologists, helping them to build up this system. It's called soil chips. I consider it as a window because soil is opaque. You can't really see them live, see how the bacteria, fungi interact with each other. So we built this microfluidic device, which is made of PDMS, transparent silicon. Using the method called photolithography. Is similar how you make the computer chips. Inside of those tiny chips, we could construct microscale features, structures like a maze, and then we inoculate fungi. We inoculate the soil samples here, and at the entries allow them to migrate into the chips. And then we can observe them in real time to see how they interact with each other. Then you may wonder, well, how many of them will go in there? And our sequencing shows there's about 30 to 40% of soil bacterial community that manage to move in there. So that's considered a lot. And then it comes with the challenging of image analysis because we put those chips under inverted microscope, and those natural soil samples, they don't have fluorescent labels, so we have to identify them manually, count manually. And it's a common practice here that we have students count them one by one, and with thousands of imaging. I will show you the image, how it looks like. So this is the maze we construct in our chips. The channel here you're looking at, they are 10 micrometer wide, and those little white dots, they are single bacteria. You could see them pair with each other like two bacteria. So there's some also little clusters here. And I consider this as quite clean an image. And those are the walls, and we construct the maze, and you can see they hide away from each other. Sometimes we have fungal hyphae in there. But then the image quality changes, like with different soil types, and we do it at different places. And we try to use simple thresholding method image. We couldn't get a very satisfying result. But when we have tried to identify by ourselves, for example, here you look at this bacterial cluster here and this fungal hyphae. We first examine the edges and the corners. Then we move to more complex features that tells us, oh, this is a bacteria, maybe this is a protist. But this idea that we're moving from simple features and then progressing to the next level complex feature, that reminds me of convolutional neural networks. So then we were considering, well, if we use the deep learning, it will give us more robust result, and we could analyze like large data. And then we tried with this famous algorithm, mask RCNN, and I guess a lot of people know about it because it's widely used in like different fields, medical, agriculture, engineering. So to test it, we got soil samples from across the world, from Greenland, Sweden, and Kenya. And it's worth to note that in Kenya, we took our chips and we buried them under the soil. After a few months, we dig them up. We use a portable microscope to look at this image. And the samples from Greenland and Sweden, we brought them to the lab here. So we have a better microscope here, which means we have very contrasting image qualities between those samples. And then we labeled our image. We have different classifications. We have single bacteria, we have dividing, which is literally two bacteria attached with each other. And we have clusters, we have structures, and also fungal hyphae. And then we use ResNet 101, and we train it on 500 imaging. And I like to mention that because the most difficult part is to organize the data set, because for the if you look at the previous image here, with different students, so we try to ask them to help us to annotate. They have different standards because some are out of focus, some are more blurry, so people have a huge variance when they give us the result. So it's really difficult to keep it like constant standard. And then after the training, we actually got pretty good result. You can see even with the image from Kenya, it's not far from the image we got in the lab here. So we are very happy. And the most important is the consistent result from deep learning is that we have manual counting. Like I mentioned before, when we have student counting, some people, they tend to count more that a little slightly out of focus because it's easy to identify. Some they consider, oh, this is maybe a bacteria, this may not. So there's a huge difference. But now we have more consistency here. This is what it looks like. So for the sample from Greenland here on the A, you could see it's more like open space. Inside here we have bacteria here, then the purple one are the structure, and the B is more like a different structure, like within the maze. And then in Sweden, and the Kenya, the image will have lower resolutions, but it has no problem identifying and counting them. So with the deep learning, we are managed to segment every single bacteria, and we are able to count them to obtain the abundance, and we can even calculate the size. And abundance. So, but it's also very interesting now we can analyze the morphology, the shape of the bacteria. So you can see the very difference in these three regions. So here what we're measuring is the major axis length and the minor length. So you could see the ratio of them to see if they are more like sphere or they're like more elongated. You can see quite different here. In Kenya, they are more like with a larger size and more tend to be the round. So this is a proof of concept we're trying to combine with our Zou chip and with the deep learning to give us more information about the Zou bacterial community, and we will moving forward to identify more different species, proteins, fungal hyphae, and everything. So now our next step, we will try with more complicated scenarios where we have a lot of soil particles, and we have more clusters, bacteria. So the key points I want to show here is now we are as the end user, so we want to show people how because in this soil microbial ecologies, it's not very common. Well, people are moving forward to adapt to this newer technology, and we want to show them with this combining soil chips and our deep learning, we could get more information. And the most important is the community characterization is mostly characterized via molecular analysis here. And there's a lack of understanding how it's correlated with the cell morphology size. So that's the direction we are moving to, because with the soil chips, it's transparent. We could look at them live and see the interaction. And when we do combining with molecular analysis, doing the sequencing, then we can also get cell morphology information, and we can get more results from that. And at the end, I would like to acknowledge the contribution from my team, the Alexandros, who contribute to the deep learning training, and our student Eric, Julia Seaman, they contribute the soil samples, they did the measurement, and our advisors, Pat and Edis, for the microfluidic and the soil ecology knowledge. All right, thank you very much.